All right, cool. Yeah, thanks for being in the talk. So again, we're going to talk about the microbiome, which in veterinary medicine in general, and I think in human medicine, it's a new buzzword. See what I did with the like buzz? So yeah, all right, let's get going. So again, thank you guys, because that project also kind of like came out of stuff that I was talking in my last lecture, so it kind of spiraled a little bit out of control. And, you know, I applied for a little bit more money. I got more money. Thank you. And so we did this. And guess what's going to happen? I'm going to reapply for more money. So yeah, but basically, why did I want to do this? So because, like I said before, right, the bee is a farm animal. It's producing food that enters, human consumption. And so just as with anything, if you abuse a drug, if you don't use it right, you're setting yourself up for failure, and that's frustrating for your doctor. That's frustrating for you because it comes back to bite you. And so, and in that case, so when I teach my students, like I always ask, like, what is rule number one? And rule number one is do no harm, right? Because we can cause a lot of harm in many different ways. So whenever I do something, I want to make sure that do I have a potential to make the situation worse? And like, yeah, we can. So that's always on my mind. And so, as I said before, since 2017, right, the government came down hard on it. And now, which sometimes I refer to as a little shotgun wedding, where beekeepers and veterinarians, you have to play nice now, And so this is why we started at education. And so if you are potentially looking for a veterinarian, and you don't know where to look, so one of the things that you can do, you can go on that site. It's a honeybee veterinary consortium. It's a bunch of veterinarians that do like bees. A lot of them are private beekeepers too. And what you could do is actually, there is like find a bee vet, and then you can you click on this and then put your zip code in, and then, maybe there is somebody in your area. But like I said, we're kind of working hard, and yeah, I do have a couple of titles behind my name and letters, but one of them that I'm really proud now is that I'm one of 9, and there's a second one, Elizabeth Mackey. Raise your hand. So Elizabeth is also certified. We both are now like 2 out of 9 in the country. So 2 out of 9 certified honeybee veterinarians. So yeah, so which is really great. And so again, we're trying to educate our colleagues that when their phone rings, that they're not going to be like, huh, what? So yeah, but it's a work in progress. So let's go right in. I got a lot of stuff to share with you, so I'm going to be motor mouth up here. So again, there's lots of peer-reviewed papers out there, and I'm going to mention a lot of papers. And what I do is I'm going to try to summarize them, because that is a hotly debated topic, and I want to make sure that I'm not just communicate an opinion. Well, clearly I have an opinion, but I try to validate my opinion with as much science data as possible. That is not just a random opinion. So we have these peer-reviewed papers, which, again, they go through a lot of scrutiny before they're being published. And so they looked at the antibiotics and what it affects on the honeybee larva, right? And so they said the results demonstrate that the concentration, I will talk about this, of antibiotics kills half of the individual's larva. So again, think about it. You go with your kid to the pediatrician, diarrhea, the doctor gives, and it could be that your kid dies overnight. Because like, I don't want to do that, right? I mean, that's crazy, but killing a lot. And so we're going to talk about this concentration there. So antibiotic exposure can negatively impact the health of the honeybees, especially tetracycline. And we're gonna go through that. And so, like again, that talk, I'm gonna talk myself out of business, because my business would be to prescribe you antibiotics. But like I want to do good. I don't want to do bad. So what is that toxicity that they mentioned? So the LC50, it's the lethal concentration 50. That means in toxicology study, you expose a population to escalating drugs, and then you want to find out that concentration that kills half of your population. So a toxicologist thinks that's cool. That's like there's a lethal dose, LD50. So basically, this is 125 micrograms per milliliter. I kind of like try to put it in a little bit more visual, everyday thing, and it's literally if you would take 2 to 3 grains of table salt and you put that in a thimble and you add water, like sugar. So and that is not a strong solution. Do we all agree? 2 or 3 grams of salt. I mean, there could be a shot, and you wouldn't even know that there's salt in it, right? It's like this is the concentration that kills half of the. And if we use, like, let's face it, if we use antibiotics, a lot of times our bees look like this afterwards, like, okay, a little bit tetracycline is good, maybe a lot of tetracycline is better. You're welcome, bee, kind of like. And you can imagine what that does to the colony, right? So the conclusion in this paper is tetracycline has a deleterious effect on honeybees' particular larva, right? That's the next generation. We are the stewards of that next generation. And, again, we are wiping out also their kind of like gut flora. And that also has a long-lasting effect, and we'll talk a little bit more about it. So, again, the treatment dose. So what is legally proved? So I'm using Canada as an example. So they're using 10 milligrams per gram, so that is really interesting. Remember what the lethal one was? 0.1 per gram. So we're already using way more to nip that bacteria in the bud. But while we are winning, we also lose a lot. We cause a lot of side effects. So basically, this is from this paper. So here you see colony-forming units. So this is how many bacteria grow. And then we're going to have oxytetracycline, tylosin, lincomycin. And you see actually, if you just use this, you're going to reduce the colony-forming unit. That means you're decreasing the population of the bacteria a little bit. But you would need to use that 100, to really make a difference. If you do 1,000, you really wipe them out, but you probably wipe out a lot of larvae too, right? So, but that's kind of interesting to me that we have recommendations out there, and we have toxicity, and those recommendations are much, much higher than toxicity. So that's a little bit weird. And again, it actually causes a lot. In human medicine, like I said, microbiota is a big thing now. And they have shown that if you would travel somewhere and you have traveling diarrhea and you're taking an antibiotic, that it literally takes years to reestablish your normal gut flora. Not like a couple of days. You may feel good, but you wiped out the really nice bacteria in your gut too, and exactly the same happening in the hive. So again, our results show that antibiotic treatment, persistent effect, right? Persistent, not just like, so basically more or less for the life of the honeybee. And then, that exposure resulted in decreased survivorship, right, in the hive and in lab experiments. So again, these papers are fairly solid papers. They're not just kind of like, I did this, I sprinkled a little bit tetracycline. So they're scientific papers. So, and again, like if you read them, they paint a very dark picture about throwing antibiotic in there. So not only potentially, maybe you get rid of this, but you're exposing them to be vulnerable to a host of all other problems. This is an interesting thing. So group 1 is the control. Group 3 had basically the antibiotic, and then again, this is like they're looking at what is the amount of bacteria they get out, and as you clearly can see, the control group has significantly more bacteria living in them, and we're killing everything out in their gut. So it's kind of like a sterile bee. If this would be a human, it would be the boy in the bubble, right? Because like everything now, like you're wiping out potentially the immune system with this. Okay, so new evidence showing that the destruction of gut bacteria could increase the vulnerability to Nosema. You probably have maybe a small load of Nosema in your hive, but the immune system keeps it in check position, keeps it kind of like, all right, there's a little bit Nosema. So now you're throwing antibiotics on there, Nosema is like, ooh, So one of the problems is you all know that the clinical sign for Nosema is diarrhea, right? And the reason why the bee develops diarrhea is because that's the replication cycle of Nosema. They enter these epithelial cells in the gut, and then they have babies. And then when they're ready to hatch, literally the epithelial lining ruptures. So if you would be a mammal or a chicken or whatever, you would have bloody diarrhea. So because they don't have red blood, they have hemolymph, so we don't see it as bloody diarrhea. But, it's like, oh, that's concerning. And so this is why these hives are nasty, because like literally Nosema destroys that lining from the inside, right? That's just the life cycle of Nosema. So that's the other group. So basically, they had a group that had Nosema, and then they had Nosema, and then they gave them antibiotics. And then the green bar is obviously fold of difference in Nosema. So you see actually the ones that just had Nosema, that bar you can barely see, has a little bit Nosema. The moment you add an antibiotic, Nosema goes to the roof. So you may have gotten rid of a bacterial problem, but now you invaded, you made it worse. Oh, thank you so much. Thank you, Madam President. Okay, cool. So, but see how crazy that is? Like, so, pick your enemy, So this is really cool. So basically, what they're looking at, so abicin is actually kind of like, it's part of the immune system of the bee. It's kind of like it's basically telling the bee, hey, our immune system needs to be upregulated. And again, the group 1 is the control one. And then look what it does to that immune modulator. It basically downregulates the immune system. And this is why you're actually seeing that rise in Nosema, right? Defensin is like, it's cool, they named something like what it does. It's like defense, of the bee. It's like an immune system. Same thing. Like you're down-regulating with the antibiotic the natural immune response of the bee to fight stuff, right? And again, this is another one. So it's not just, it doesn't affect one factor, it affects the whole immune system, which is really scary. All right, so the conclusions say that not only leads to complex problems with antibiotic resistance, but it also can impact the honeybee directly. So you get the idea that you're suppressing the immune system, so which is crazy, which then obviously not, the honeybee has a non-adaptive immune system. That means like, mammals and stuff like this, we create antibodies, that's why we get vaccine for specific diseases. Their immune system is a little bit more, let's say, primitive, to lack for a better word. And so once you downregulate their immune system, they're going to be susceptible to a whole slew of problems, right? Virus, Nosema, bacteria. So you're kind of like doing a dance with the devil here a little bit because you're really causing problems. But also it affects their behavior. So you're literally, it's like probably it would be kind of like maybe they're feeling like they just smoked a joint. They're like, I'm not showing up for work, like, and where is the flower anyway? I forgot, kind of like in human medicine, there's a lot of talk about, you probably have heard it, the gut-brain axis, right? They recently have found out that actually if you combine all the neurons that are in your gut and you add them up, it's exactly the same amount of neurons that we have in the brain. Which is really crazy. So that the grammars are saying, I don't know, it's a gut feeling. Well, it turns out it's really true because your gut talks to you. Listen. So, and now they're finding that out with the bee, the same thing, right? There is this connection between like the brain and the gut. So again, beekeepers may inadvertently make the situation worse by administering antibiotics because they're altering the behavior too. So this longitudinal stuff, right? Fast developing bees, I thought, then it's a problem. And this is the problem. They actually die outside of the colony. So you don't find those bees. And that makes it fairly difficult to do research when the body is not being found, right? But this is kind of like the colony dwindles away. Yeah, because that poor little bee never made it home. How do you do research on that? But, this has been shown in the lab. So again, many papers, right? So, and this is a big problem. So metagenomics is a big keyword now, where kind of like you look at all the bacteria that live in the gut and everything, and they see a high residence of tetracycline resistance. And it's scary. The more people look at this. And again, it's a food-producing animal, so it's kind of really close to the food chain, and those bugs could potentially, you ingest that potentially. It's like, do you really want to have these highly resistant bacterial genes in you? Like you do. This is from that paper, which is amazing. So basically, the higher the bar, the more genes that they find that are antibiotic resistant. Okay, so So it's basically the amount of how many genes do I have that are resistant to antibiotic. And you see Connecticut and Washington, this was their sample sites, okay? Recently established from commercial packages. All right, let's see this. They didn't use antibiotics in those states for 2 years. Oh, look what's happened. They're slowly but surely establishing a little bit more normal stuff, right? Oh, for 25 years. So it takes a really long time, right? We said it has a very longitudinal problem. Obviously longitudinal not for the individual bee, because the only individual bee, like 2 months or whatever, and then the new, but it stays in the hive, and that genetic material is being passed on. That's hanging on. So still 25 years. Versus if you look at the rest of the world, Switzerland, the Czech Republic, and New Zealand, nearly no, because in those countries it's illegal to use antibiotics. And that's what I've been saying in the United States. You know, you guys have the luxury to potentially have access to antibiotics. If you're abusing it, it's going to be a European situation where it's not going to be. So anyway, this is why you have to use that very responsibly, because otherwise they'll take that away from you, right? Which maybe not a bad idea. So again, so what do we know? So it kills, like, you know, we have an LD50 established. Again, it's causing maybe even more problems in an Abel's nosema. You know, their behaviors, they didn't even know where the flower is anymore, right? And then these long-term you know, exposures also cause the immune system to be defunct. So a lot of problems. What is the other options? Like I said, sometimes you have to. So yeah, this is my idea of me and my student working on hives. So how do we deal with this? You know, there needs to be something, because like I said, I don't want to stand here and say you should never ever use, right? Because also my grandma says, whenever is like never or ever is always wrong. You know, like there's always an exception. It's like, Grandma, thank you. You're so wise. And so anyway, papers are coming out, lactobacillus. So it helps actually with that. So it attenuates these negative things, all these negative things. So you can help your bees out with feeding them probiotics, you know, which is really cool. So it's beneficial. So in that thing, so IX3 or LX3 or whatever, so this was just a commercial probiotic, but they basically, you know, it clears actually, even Binnie bacillus larvae, even American foul brood can be cleared with probiotics, which is actually great news. All right. So again, the combination with oxytetracycline and the probiotics suppressed Binnie bacillus larvae, American foul brood, more effectively than oxytetracycline alone. Right? Because you know that oxytetracycline alone really hammers the immune system. So therefore, we have to offer the bees kind of like a new population that helps with the immune system. And that's where your probiotics come in, okay? This is my talk about the microbiome. So again, I always, when I'm going down a rabbit hole or whatever, so I'm always like, what's happening in human medicine? You know, is it just me being crazy? So, and if you see this is basically a search database where you can put something in. I put in probiotics, and it mainly brings up human papers, scientific papers. But you see in the recent years, this is the definition of exponential growth. So, and I'm always saying, it's like, if this would be snake oil, if this would be something you do not see interest, especially in high funding, NIH funding, like lots of thousands, maybe millions of dollars for snake oil, it's usually kind of like you do a study, you have surprising results, positive, surprising, and then you do another study, you apply for more grants, and you see then suddenly it goes around the community, and there is like, you know, buzzwords. And so I think, wow, you know, it's like there is probably something to it. Let's dig into this. And we know a lot about these probiotic supplements. You see this little thing with the mucus layer, and we know this is really important, and this is what potentially also kills these bees to European foul brood and American foul brood, because that mucus layer is destroyed, and then as you see, there's bad bacteria, and then they colonize this. So the mucus layer, I want to say, is a little bit an equivalent to suntan lotion, right? So it's like, so it protects the inner epithelium from these bad bacteria. So and so a lot of time, what European foul brood or American foul brood, it gets rid of that layer. So and then exactly, you'll see exactly what's happening here, and then you have those bad things eating their way through, and then you have death. All right. So again, this is one of those fancy slides, and you see very little. I don't know the pointers. I don't know how to work the point on this thing, and I don't want to push it and lose it again. But you see a little bit. You see a little bit the slime layer here. See, this is this protective slime layer, and you see up there how it's nearly gone or something like this. So this is potentially getting whittled down, and this is a sick larva. Again, so there's lots of ways how to look for success with it. And I always say a very objective way to look if an animal is doing well or not is weight gain. So that's like, oh, it's gaining weight. It probably is eating and doing well. So here I'm going to start to talk a little bit about, because there is some controversial stuff out there, and people say, well, there's not a lot of data on that probiotics are really good. And I'm like, really? No, there is. So how do you conduct your research is really important. And so one of the things that I find, like it's a trap, if I feed lactobacillus to my bee, and I wait 4 days, and then I dissect my bee out, and I look in the GI tract, and if I don't find lactobacillus in the GI tract, I cannot come to the conclusion that, oh, it didn't do anything. So it's kind of like if I take a vitamin D3 pill, or whatever, vitamin pill, and the doctor, I'm going to go to my doctors, and it's like, would you mind peeing in that cup and pooping in the cup? And they're going to look at it, it's like, I don't find any vitamin D in your pee or in your poop. I don't think vitamin D does anything. I'm like, second opinion, right? Because it's that whole rat tail. It's that whole rat tail that's happening. These metabolites, what I showed you, this immune system, that's what we need to look. Like I said, this is from human medicine, and it's very similar though. And you see these probiotics, they go into the cell and see what they release, plasma cells, and this is interferons, TH cells, these are T helper cells. They're really helping the immune system. So again, just because I wouldn't find lactobacillus, and maybe it only takes a day or something like this to activate, and then on day 3 that lactobacillus is dead and I can't find it. But if I would check for tumor necrosis factor or those TH cells, they have been upregulated, right? So I'm like, okay, it's really important in how we look at this. So again, their conclusion is that the gut microbiota of young bee promote weight gain, stuff, and genes are being switched on. Genes that are dormant are literally being switched on, which is really powerful in my opinion. And so again, what they're already looking is like there's actually a fairly lot similarity between the microbiome of the honeybee and human research. So that gets them excited. So, and that's the whole concept of One Health, right? So we can potentially improve human medical science with honeybee science, and that's to me as a veterinarian, that's a dream. It's like I can really maybe contribute to human health doing my bee stuff. Whoa, that's so cool. So again, there's some mechanisms on how that works. So a lot of people try to explain this, and there's lots of different data out there. So again, I just want to point out that we have a lot of good peer-reviewed scientific evidence that there is benefit to it. All right, sometimes I know that we thrive on sensationalism, and sometimes, it's like there is one paper that comes out and says it didn't do anything, and then everybody focuses in. But I want you to understand there's also probably 10 or 100 papers that prove the opposite, and that's science, okay? So this is really cool. And it's not just one paper. Look at all these papers and what their conclusion is. And a lot of them, again, we talked about this morning, in vivo means they actually do it in the bee. So it's that real complex mechanism, and in vitro is kind of like in a petri dish. But you see there's a lot of isolated lactic acid bacteria, and then what it does, it inhibits Melissiococcus. So it's not just like one person's opinion from one lab. These are all different researchers, probably even from different continents, coming to a very similar conclusion that I think the evidence is so strong we shouldn't ignore. All right, here's a couple of examples that I pulled out, which I think is really cool. There's Bacillus subtilis. It inhibits a Penibacillus larvae, American foulbrood, and actually inhibits a fungus, the fungus that causes chalk brood, because it produces its own fungicide. Isn't that cool? Yeah, right? I sense excitement. I can see it. Yeah. Okay, and like it inhibits nosema too, right? More lactic acid produce like this organic acid, hydrogen peroxide. We all love hydrogen peroxide, right? I think Dr. Bartlett says this is how bees wash their hands, right? So yeah, that's his quote, I think. So and so they produce all these compounds. So again, if your research is just like I couldn't find lactobacillus, therefore it doesn't do anything, I'm like, I don't know. Right, and then bifidobacterium has been shown to enhance the immune system, and so actually bifidobacterium is really cool because I'm going to show you in a second. It actually, there we go. It produces acetic acid, lactic acid, and even some of those produce formic acid. It produces formic acid inside the bee. Isn't that cool? And that's what we want. Oh my God. Come on, you guys. Yeah. That's it. Yeah. You guys all would be good in Vegas. You're good poker players. You're like, I'm excited, but I won't show it to him. I'm excited. Man, gosh, look at this. This is like, and again, it's like I have the papers there to show it. It's not my opinion, so it's kind of like even during winter. When do we lose our bees? In winter. So if we feed them maybe probiotic before winter, maybe we're I always say to the students, give them the yogurt, feed them the yogurt. So, kimchi. I love kimchi. Every day I eat kimchi because I believe in fermented products. And so these guys actually fed like kimchi. And think about it. What do we know about pollen once it's in there? What is it? Bee bread, right? And what is bee bread? It's fermented pollen, right? Are you connecting the dots? The bee's trying to tell us something. I like lactic acid. I want that because it's good for my immune system, right? But be careful. Be careful. There's evil bacteria amongst the good one, right? So they found actually that not just any lactobacillus, you can actually cause harm if you don't use the right one. So this is why it's really important that if you buy probiotics or whatever, that you maybe probe a little bit and say to the people, it's like, hey, did you do some research? Or did you just like order a little bit lactobacillus and mix it in? And, so it's like with anything, it's like rule number one. What is rule number one? Oh, God, you're so good. All right. So novel lactic acid bacteria inhibits Pinibacillus in Bribus. So again, what I'm trying to point out is like lots and lots of peer-reviewed data out there that literally like these good bacteria are actually killing the bad ones actively, either the bacteria themselves or their byproduct. All right? Really cool stuff. So the big picture is, I think they're inducing the immune system because with that gut, also with the gut-brain axis and stuff, they have the systemic, you're upregulating this. So it's kind of like in winter, you already say eat your vitamin C drops or whatever, right? So upregulate your immune system, vitamin C. This is like upregulate your bee's immune system with the probiotics, okay? Probiotics are recognized to enhance bee immunity, enhanced resistance to disease, wax glands, so you're going to get beautiful wax. And maybe you're going to win the wax show, and increased honey production. So basically, because I always say, when people say, yeah, but that's going to cost a lot of money. It doesn't cost you money. It saves you money, right? Because if you spend $50 on the probiotics, and let's say you believe that, and potentially that colony overwinters, what is the other option? You save the $50 and you spend $200 on a new nuke. Right? So think of it not an expense, think of an investment, okay? Lots of publications. Again, in vivo, in vitro publications. I just want to show you that this is a little bit overlooked in my opinion. There's a lot of good scientific data out there, production of lactic acid. So, and they all show positive outcome, okay? Again, with neurological process, like I said, that it's exactly the opposite of what the antibiotic does to the bee. Like I said, the antibiotic, the bee kind of like, I have no idea, what does the flower look like? I don't know. Kind of like, it actually, if you destroy their gut microbes, it has this, again, think about that gut-brain axis. So versus, like you keep that healthy and they're being sharper. This is crazy. I would have never gotten this right on a multiple choice test. Their data revealed that over 700 genes are being switched on with these good bacteria. Do you remember, has anybody pattern recognition? Gallionella, Bifidobacterium, Snodgrassella, these are all bacteria that I mentioned earlier in our MyDog test, that once we supply those, we can actually culture those out. These are the good antibiotics. And what do? They turn on genetics. I mean, it's crazy. And again, Grandma was right. Grandma said, you are what you eat. Right? You all have heard that. You are what you eat. Hello? Grandma, gosh, I always say to my students, respect your elders. They know shit. They don't know the science, but like sometimes we don't know the science, but don't ignore that stuff, okay? So again, host learning and memory behaviors. That's exactly what you want to be. You want the bee to come back with a waggle dance, and you want it to be the most beautiful waggle dance, because if it's 2 miles away, it would be kind of like she flies back and she's like, oh man, was that on the 3rd tulip poplar, right? Or Or was that over 85? I waggle a little bit. No, you want them to like, no, this is the coolest thing. Take exit 85, and then there's a McDonald's right there, and that's how, and so it helps them. And then obviously memory function, and they express this and communicate this, okay? So again, physiology, immune response, again, I think it's more complex than we currently know or understand. And all these articles are fairly new articles. They're coming out in the fairly recent years, and so we're just scratching the surface. So putting it all together, I think, the hemocytes is basically kind of like 10 minutes. Thank you. I think I could do this in 10 minutes. So yeah, and so basically, it's well tolerated. So if, like I said, there is maybe some lactobacillus that is not that good as I showed, but like the commercial stuff that has been doing well and where people like, so somebody uses probiotic, ask them what their experience is. I've yet to find a really good publication that says, oh, we kind of overdid it with those probiotics. Ooh, So, but I think you would maybe have to bury the larvae in it so they can't breathe. I mean, I don't know. Like I said, everything is dose dependent, kind of. So you could, but if you use it kind of like in a more or less common sense way, I have yet to find kind of like really good evidence that will make something worse, right? So the data speaks actually for like please use it more or less, and it has very little risk, has nearly no side effects, okay? It contributes to the overall well-being of the honeybee, improve the immune system and gut health. That's what we want. Again, I'm excited about this because it's like now in human medicine, so these are all important kind of publications, disease modeling, honeybee as a model for gut microbiome, because, they can raise them and it's, I don't want to say cheap, but it's cost effective to keep those in a lab. You have a fast turnover, right? You can collect a lot of data in one summer. So that's pretty cool. So I'm really excited. So yeah. So I hopefully the MD is getting excited about that, and we'll learn more about the honeybee. And the more we learn about the honeybee, the more we know about our gut microbiome. And I think a lot of them is very translational. So again, I think we have a lot of different reasons why, in my opinion, we should be feeding probiotics. And so my students know already, like after every inspection, I'm kind of like, did they get the yogurt, right? And we sprinkle a little bit over it, and I really feel that my girls like that. So like I said, let's keep it real. Sometimes you need antibiotics, especially in commercial operations, maybe, I don't know. But again, please think of the do no harm thing. And if you use antibiotics, please at the same time use probiotics, right? Because you're wiping out these things, and you can say, okay, maybe I'm going to use antibiotics on Monday or Tuesday to give some time to clean up the bad boys. But then maybe on Wednesday you should be out feeding probiotics, right? Maybe you want to have that little bit of lag behind, and then you want to re-establish these good bacteria that you obviously destroyed. And you want to upregulate the immune system because you did downregulate, yeah? And you didn't mean to, but that's a side effect, okay? So again, I think, I don't think we should ignore that. There's plenty of good evidence out there, and again, probiotics in sports, they keep feeding it to athletes. Hopefully the UGA team ate their yogurt this morning when they now fight Tennessee, kind of like, hopefully they're strong. And so, but there's a lot of evidence out there, and I always say, just because we don't understand the whole thing yet, just because we can't explain everything, because let's face it, it's very complex mechanisms. And there's hardcore scientists out there. They're like, all right, that's nice that you have that your bees survive over winter. Explain it to me. And I'm like, I don't know, I can't explain it. I'm like, I don't believe it then. But the bees are alive. I don't believe it. This is not a belief. They are alive. But there's people that want to have everything explained, and then you're going to disappoint them. So I'm one of these bad researchers that I'm like, I just look at results and then try to look back and and if I can't explain it, I mean, my favorite example is Einstein. What made him famous? Relativity theory. Just, I think it was 5 years ago, literally, where it went through the news that they had their first proof that Einstein was really right. because they measured something out in space and they're like, oh my God, we have, but I mean, that's my point, is like, as long as it's logically thought through, as long as you have results, just because you can't explain everything doesn't mean it's not working. So that's kind of like how I roll as a kind of scientist. So anyway, I think my time is up. Thank you very much. I've got 5 minutes. Yeah, these are pictures that, because I don't have a life, this is pictures that I do on my spare time. So in the beginning, like, I was doing this and my wife was like, what the hell are you bringing dead insects in the house? I'm like, hear me out, you know. And she's like, well, I guess I married him. And now she brings me dead insects. She's like, hey, like, what about this? I found a scorpion. Do you want to do something with this? I'm like, oh yeah. So anyway, if you like any of this, and this is why we named that conference One Health and the Honeybee, because this is like that apitherapy thing, because I think, you know, One Health is a big buzzword where, you know, everything's interconnected. I mean, we all like to cite, if the honeybee dies, you know, population decline, humans die too. And I'm like, we're all connected. And so I think, you know, in a positive way, as like the science that we can get from the honeybee can be beneficial. And so thank you guys very much. And I think my time is up.